methods. Experimental design in the study consisted of HYGMS's culture using the explained primary culture method, which obtained from the umbilical cord. Cells were maintained using the MEM, 10% of FBS, 1% of antibiotic and antimycotic. And furthermore, the master micropatterning was made from silicone material, followed by molding the agarose. And this study used two micropatterning methods, agarose as a substrate and agarose as a stem, which will be explained in the next slide. And then cells were then seeded on a micropattern to observe their viability using MTT assay, their attachment using SEM imaging, and the cell pattern using ICC against DP staining. Okay, this is the method to make a micropattern. First, after the silicone master was made with a line pattern with the size of 500 micrometer and 1000 micrometer in this study, agarose was molded on it. And then for the AMSU method, the protein coating is was made on the grooves of the micropattern. And then for the AMST method, the coating fibronectin is on the trace of the micropattern, then stamping it on the cover slip. As a result, the desired pattern was formed over the cover slip. And then result and discussion. On the figure 1, we can see the agarose micropatterns that used in this study with the size of 500 micrometer in figure A and 1000 micrometer in figure B. And then the next result is cell viability. Cell viability on IMSU and IMST show that IMSU have a higher OD value compared to controls without a pattern. Also on IMST, which had a higher OD value than the control. And the both 500 micrometer and 1000 micrometer IMST show higher observance than the control. As we can see here, we can also conclude that the micropattern substrate was not toxic to HWG MSCs. And then cell attachment. We can see here in figure 3 and figure 4, cells can attach to both of IMSU and IMST method. On IMSU, cells tend to not spread but are rounded. But in IMST, cells can spread well. This was related to the substrate stiffness, which indicate that cells tend to be rounded on soft substrate rather than on the rigid substrate. And also, on IMSU, cells not adhere well to the substrate. It's because the lack of cell adhesion to the substrate. And this is related to the successful attachment of the fibronectin as an ECM that attaches cell to IMSU. Several studies have stated that protein grafting onto agarose was difficult to achieve, and major chemical modification have been required for successful results, such as using UV irradiation process. Because of the IMST method, it shows a better attachment, so it's followed by observing the growth pattern on IMST using ICC. On this figure, we can see that cells on IMST were seen to form a line pattern of 500 micrometer and 1000 micrometer in size. According to Ekman et al. 2012, chondrogenesis is highly dependent on cell density, so the micropatterning with IMST can be a promising method. So this is the last section of my presentation, the conclusion. Agarose gel becomes a polymer that can be used in micropatterning application. And the agarose micropatterning stem method was able to support the attachment of HWG MSCs better than agarose micropatterning as the substrate method. However, further research is needed to determine the effectiveness of AMST in directing HWG MSCs to become chondrocytes for a longer period of culturing. Okay, thank you very much for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you for the nice talk, Ms. Jinda Hania. So we will go directly to the third talk or the last talk for this session. Please take your time, Ms. Anggia Prastyo Putri. You can share your screen if you want to present yourself, or do you need help from the organizing committee?
No, thanks. I'll present my screen myself. Thank you.
Okay. Yeah.
Okay, good afternoon, everyone. Assalamualaikum warahmatullahi wabarakatuh. So thank you for this opportunity to present a sneak peek of what I've done in my PhD. Just a very small part of the theory behind the work. So I will present today an insight into the Seesaw Effect, which is really a good observation that has been noticed in the clinics where there is differences between susceptibilities to different classes of antibiotics in MRSA. So MRSA is methicillin-resistant Staph aureus. I'm sure we're all familiar with these pathogens, and the methicillin resistance is caused by an acquisition of the MET-A genes that encodes for penicillin binding proteins 2A, which means that the methicillin antibiotic can no longer have activity to kill the Staph aureus bacteria. And therefore, this MRSA is also resistant to all beta-lactams class of antibiotics, except for ceftaroline. And the mainstay treatment is vancomycin, but of course, resistance to vancomycin is increasing, and so is resistance to other antibiotics classes as well. So basically, like I mentioned just now, that MRSA, sorry, the treatment for MRSA is mainly with the glycopeptide vancomycin, and teicoplanin is used, but not very often. And there are other classes of antibiotics that can be used to treat this pathogen, including daptomycin, which is a lipopeptide, and linezolid, which is an oxazolidinone. So this daptomycin targets the cell membrane, linezolid targets the protein synthesis. And there are also a few other antibiotics, including ceftaroline and tigecycline. And these three, telavancin, oritavancin, dalbavancin, are three of the newer generations of antibiotics. But today I'm going to talk about the two, vancomycin and daptomycin, because they are the ones that are mainly used to treat bacteremia caused by MRSA in the clinic. So vancomycin itself was discovered in the 1950s, around 1958. But its use in the clinic was not really used as often as other antibiotics that have been discovered after that, so that the resistance to vancomycin didn't really emerge until the 1980s. Whereas daptomycin, it was introduced in the early 2000s, I think it was 2003, but resistance emerged as early as 2006. So it's really important to make sure that due to the limited arsenals that we have, that we find ways to ensure that we can use these antibiotics as long as possible. So the focus of today's talk was on the different targets of these antibiotics. So if this is the gram-positive bacterium cell wall, so the vancomycin targets the peptidoglycan synthesis process by targeting the lipid 2, and the daptomycin targets the cell membrane, and the beta-lactams target the penicillin binding proteins. So this is basically just providing a sort of an overview of the different targets that they access within the bacterium cell wall to kill and inhibit the growth. But the vancomycin resistance emerged around 2000. So the emergence of vancomycin resistant Staph aureus, hence the VRSA, it's caused by an acquisition of a FAN-A gene, which causes a change in the binding site of the vancomycin from the ALA to D-LAC. Therefore, Staph aureus strains that have these genes and this mutation will be resistant to vancomycin. But the occurrence of vancomycin resistance is very rare. what's even more concerning is the fact that there's this visa or vancomycin intermediate Staph aureus, which has not resistant fully, but the MIC or the minimum inhibitory concentration increased from 2 to about 4 to 8. And this causes a lot of problems in the clinic because the clinical outcomes are not well. A lot of cases lead to sepsis and also death. And for daptomycin, there are also different mechanisms of resistance, particularly these are ones which is mutations in the NPRF genes and RPOB and so on. So what happens is this is daptomycin is a cationic antibiotic that is attracted to the negatively charged cell membrane. So the mutations in the NPRF genes cause the cell membrane's positive charge to be increased. So it kind of repels the positively charged daptomycin. So therefore, so the seesaw effect is when we see these Staph aureus strains that have resistance to vancomycin or daptomycin, but then it turns out the same strains actually become more susceptible to beta-lactams. So like I said in the earlier part of the MRSA are resistant to essentially almost all beta-lactams, but those that have resistance to vancomycin and daptomycin, turns out they actually became susceptible again to beta-lactams. And these are the some of the beta-lactam antibiotics that have been used in the clinic and have been shown to have good susceptibility, that Staph aureus has been shown to have good susceptibility to when it comes to the van and dap resistance. So the seesaw effect has been taken advantage of by combining these antibiotics to treat Staph aureus infections that have these resistance. So a number of in vitro and vivo studies have mentioned that results that combination of beta-lactams can actually provide advantages that it can resolve persistent infections, and it allows for a dose sparing, so a lower dose of antibiotics. And also in vitro, it has shown that it can actually delay the resistance development to these two antibiotics. So there are a few proposed modes of action. One would be that the beta-lactams reduce cell wall thickness, allowing vancomycin access to the lipid-2 target. So resistance to vancomycin, especially the visa, the intermediate resistance, usually is signified by or marked by a thickening of the cell wall. So there are a lot of decoy targets that makes it hard for the
just to remind you that your time is almost finished. Maybe you can make it a bit faster. Thank you.
Yeah, sure. So that's the one. And then two, the beta-lactams increase the negative charge of the cell membrane. Therefore, daptomycin can be attracted more to the cell membrane and be able to kill the bacteria more. And also there's two more proposed mode of action. That means that the beta-lactams have the synergistic effect to target the penicillin binding proteins. And also the last one, that the mutations affecting NPRF can actually lead to decreased PBP2A production, which means there's more targets for the beta-lactams to be active. And that's just that. So anyway, basically the conclusion is that synergy is evident between vancomycin, daptomycin, and certain beta-lactams. So therefore, this synergy can be exploited to treat infections that caused by caused by Staph aureus resistant to vancomycin, daptomycin. So more clinical data is needed because there's been only limited data in terms of its efficacy. Thank you so much.
Thank you for the nice talk, Ms. Anggia. So we will go to the discussion part or discussion session. Any one of you, if you have any questions, just raise your hand and then we can give you time to ask. Any questions? Maybe while waiting, I also have one question for every presenter. For Mr. Andri Wardiana, I was very interested to the antibody specific for treating the cancer. So what I am asking or wondering is about, do you have any explanation about why the in vivo data and the in vitro data is contradictory or inconsistent? Maybe do you have any speculation? And for the second presenter, or Ms. Cinda, I'm wondering about, because you presented the data for the viability, I'm wondering why the treated cells with the agarose has a higher viability compared to the control. And for Ms. Anggia, I was also playing with the Staphylococcus in my PhD time, so I'm also very interested in your thoughts. From the proposed mechanism that you showed us, which one is the most likely or most probable explanation for the seesaw effect, if you have any explanation? Thank you. Maybe starting from Mr. Andri Wardiana, and then the second will be Ms. Dinda, and then the last will be Ms. Anggia.
Okay, thanks for the question. Yeah, I think because in the biological system is very different. So I think because we use the xenograft mice models, and I think this is the artificial tumor, right? So maybe it has, you know, it's very leaky. It's very leaky. The tumor is very leaky. So even the unconjugated one can easily go through the tumor. So maybe that one the reason.
Okay. So is it possible that the PSMA, the prostate specific, I don't know. Yeah, is it also expressed in other organs? Maybe it's not really that specific, or
yeah, in other organs. Yeah, but it's up. I mean, the expression is not really high. So yeah. So generally in the prostate cancer, it's very high, like 10 to 100-fold compared to the other organs. Yeah.
Maybe. Thank you for the answer. And we go to the second presenter, Ms. Dinda.
So the question is about viability, right?
Yeah.
Yeah. The cell viability is dipengaruhi oleh cell-to-cell contact. So maybe if we use the micropattern, cell impulse to define like a pattern that we want, so the cell is more like berdekatan gitu ya Pak, I think, because of that. So the cell viability is higher.
Makes the cell-to-cell contact is higher, so the cell proliferation is also faster. Okay. Thank you for the answer. Okay. And for Ms. Anggia, you can take your time to answer my question. Yeah.
Okay. Thank you for the question. So from the literature itself, I think that the most often mentioned proposed mechanism of action is the targeting of the PBP1 protein by the beta-lactams, and also the reduced expression of the PBP2E protein, the MECA gene reducing the PBP2E. So I think, interestingly, only PBP1 targeting antibiotics are actually more active in the seesaw effect. So the fact that the MICs against those antibiotics are much lower compared to other other antibiotics that target the PBP2 or PBP4. So that would be the main mechanism that are seen most often. Yeah.
Thank you for the answer, because I think it's quite interesting, because the vancomycin and the beta-lactam, both of them are working in the cell wall division. So if they have a different mechanism, then it will be interesting to apply or implement it in the medic. Thank you for the answer.